So today what we're going to talk about is we're going to solve some problems about translation. So translation is the process of taking an mRNA sequence and converting it into a protein. So this would be considered part of the central dogma, or the flow of genetic information from DNA to RNA to protein. Translations that how we take that RNA that was transcribed from the DNA and translate it or use it to know which amino acid sequence to put together to make our protein. Now, the problem that we have here today is we're going to take a piece of, we have this this double-stranded piece of DNA that we need to identify what's called the open reading frame. So the open reading frame is, once we transcribe our mRNA from our DNA, the open reading frame will be the region that actually encodes for the protein sequence, so it would be a start to stop codon. Now, when we do that, when we transcribe, we're going to have an initial transcript that then gets processed. And so we're going to do some chemical modifications to that initial mRNA that will modify it to get it ready for translation. And then one of the things that this particular worksheet includes is UTRs, or untranslated regions. And so if we have an mRNA sequence that's been transcribed, only a portion of it actually encodes for the amino acids or for the protein, and the sections on either side of that open reading frame is what are called UTRs, or untranslated regions. And so those are retained in our final mRNA product. They're just not converted into a protein. And so in this case, this example, we're told that the UTRs are 4 base pairs long. This is a hypothetical situation in which UTRs are going to vary in length significantly. There often could be several hundred base pairs in size, something that we couldn't depict realistically in a worksheet like this. They're more for illustration purposes. Similarly, the intronic sequences are depicted by lowercase letters here. So introns, when we transcribe our DNA, we're going to transcribe through a large section of DNA, and part of that will get removed. So the part that's retained or kept in the final mRNA, or the mature mRNA, are our exonic sequences, and the pieces that get cut out are the intronic sequences. And so in this example, intronic sequences are depicted with lowercase letters. And again, this is for illustration purposes, because these lowercase letters are, to do an intronic sequences correctly, the sequence that is recognized by the spliceosome, or the protein complex that does the splicing or cuts out the intron, is a little more complex than what we can really depict realistically in one of these worksheets. So it's, in reality, in real life, and in actual organisms, the intronic sequences are often much larger than this. They have specific sequences at both the 5' end and the 3' end that tell the spliceosome to cut here and cut here. But for this example, we're just going to use it as a way to illustrate the process of how we're going to have a primary transcript, and then how we process that primary transcript into a mature mRNA, and then we'll take that mRNA and translate it into its corresponding amino acid code. Now, when we are doing this, what we want to do is we're going to look in the DNA sequence, and we're going to read the DNA in a 5' to 3' direction. Always in a 5' to 3' direction. So we're going to look for the open reading frame, or the start to stop codon, starting at the 5' end of the DNA, and we're going to look going 5' to 3'. Now, the strand that encodes for the RNA, you can use either strand as a template. And so the open reading frame could be in the bottom strand, but we're used to reading left to right. And what we don't want to do is just read left to right. We always want to start at the 5 and go to the 3. And so if we read through this top strand and don't identify an open reading frame, or even if we do identify an open reading frame, and we want to look for an open reading frame in the bottom strand, we have to start on the right side of this strand, because that's where we have a 5. And then we read to the left for the 3. Now, when we're reading 5' to 3', what we're looking for is our start and stop codons. And so remember, when we look at our codon table, AUG, or methionine, is our start codon, and then UAA, UAG, or UGA are our stop codons. Now, here we have U's. When we look over in the DNA, we have T's. So you need to remember that when we're looking at the DNA sequence, anywhere that we have a U in our codon table, it actually will be a T in the DNA. And so what we're really looking for in the DNA is an ATG. And then we're going to move 3 nucleotides, 3 nucleotides, 3 nucleotides, 3 nucleotides, and on and on until we either see a TAA, a TAG, or a TGA. Okay? And then that will identify our open reading frame. So let's take a look at this problem and see if we can find any open reading frames here. All right, so now, here we have, it contains a small peptide. In this organism, the UTRs are 4 base pairs long. So the first part of the problem is to identify the template and the coding strands for the longest open reading frame. So what we want to do is identify any open reading frames that might be in this sequence, and then we'll figure out which one is the longest, and then that's the one we're going to work with to start off with. And so to do that, what we want to do is we want to start looking for an ATG in the 5' to 3' direction, and we keep looking at the ATC, ATG. So here's an ATG, and so this is a potential start codon. Now we don't know if it's a start codon until we've actually looked 3 nucleotides at a time and found a stop codon in frame, meaning in a non-overlapping 3 nucleotide way. And so here we have ATG, and so then we'd go TCC, TGG, TT. Now the lowercase g, that's going to get cut out. So that's an intron. That's not going to be included in the open reading frame, or in the mature mRNA, that will get cut out, therefore not be in the open reading frame. And so we go TT, and they get the third letter. We have to look on the other side of the intron, which we see right there, TTA, GCA, CGA, and then TAA. And if we remember from our codon table, that TAA would be in an RNA form, would be UAA, so that's a stop codon right there. Okay, so that's one open reading frame. So let's keep on looking in this segment of DNA, see if we can find any other open reading frames. Now, we can keep on looking in this top strand, and we don't see any other ATGs, so we can look in the bottom strand. So in the bottom strand, like I said, we want to go from the 5' to 3' direction, so we can start looking, and we find, now here's an ATG, but that ATG is going from the 3' to 5' direction. So it starts at the 3' end and goes to the 5' end, so that is not going to be a start codon. We want to keep going in this direction until we find an ATG. And then we keep on looking, and here's an ATG right there. So that is an ATG. Let's see if we have another open reading frame. If we don't have in frame, meaning 3 nucleotides at a time, another stop codon, then that ATG is just an ATG. It's not an actual open reading frame. And so here we can go CCA, GTC, TTA, and then TAG. And if we, again, look at our codon table, TAG, remember we're reading from the right to the left now, would be a UAG, and so that is another stop codon. So that is another open reading frame, or would encode for another protein sequence. And so now the question is asking us to identify the template and coding strands for the longest open reading frame. So here we have a 1, 2, 3, 4, 5, 6, 7 codons in the top open reading frame. And we have 1, 2, 3, 4, 5 in the bottom open reading frame. So the top open reading frame is the longest, so that's the one that we're going to refer to. So now we want to identify the template and the coding strands. And so the easiest way to do this is the coding strand is the strand that has the open reading frame, and it's coding for what will become the mRNA. And so when you're looking at this and you identify your open reading frame, that's your coding strand. The other strand is going to be being used by RNA polymerase as the template strand to know what nucleotide to assemble and string together to get us our mRNA. But one important thing to remember is the sequence that we find in the top strand, or in this case in the coding strand, which is the top strand, is the sequence that will become the RNA. Only T's will become U's. Okay, so now, so this top strand, this is what we're going to refer to as the coding strand. The bottom strand is the template strand. Now, the next question is write the primary transcript that would be transcribed from this gene sequence, circle the exons, and circle what the exons would be. Okay, so now remember that we're told that in this hypothetical organism, they got 4 base pair UTRs, again, just for illustration purposes. But what that means is whatever sequence we've identified as our open reading frame, the UTRs would be in the RNA itself, but outside of that open reading frame. And so if we have our start codon right here, we're going to move 1, 2, 3, 4. That's where the RNA is actually going to begin. And then if this is our stop codon, we're going to move 1, 2, 3, 4. That is where the RNA is going to stop. Okay. So now what is the primary transcript? So the primary transcript is going to be the initial transcript that comes off of the DNA. Remember that initial transcript is not going to be processed. So we don't add our G-cap yet. We don't add our poly A tail. The introns are going to be intact. So what we want to do is just copy this coding strand and change any T's that we have to use, and that's going to be our initial or the primary transcript that we find. And remember to include your direction for the RNA. You always want to include some directionality in terms of 5' and 3' ends. So we'd go 5' A U C U A U G. Okay, so I'm up to this point. Now we go UCC, UCC, UGG, UU. Now the intron sequences are going to be spliced out, but in the initial transcript they're there, so we need to include them. So then we'd go G, U, C, G, U, A, G. And now we're at the end of the intron, and we can pick up with that A there. So we'd go A, G, C, A, C, G, A, and then U, A, and C, U, A, 3'. Okay, that's a 3. So now that's our primary transcript. And now the next part of the question is asking, write the mature mRNA sequence that would result from processing this RNA transcript, include processing events. And so remember what our processing events are, is we're going to, as soon as we begin transcribing that RNA, we're going to add a guanine cap. And so that guanine, it's a G cap that also gets methyl groups, CH3s, added onto it. So it's often referred to as a methyl G cap. That's going to go on the 5' end of the RNA. At the 3' end, that's where we're going to cleave and then add our polyadenylation signal, that will cleave at the polyadenylation signal and add our poly A tail, and then the intron will be spliced out. And so what we would put is we can say have a methyl G cap on the 5' end. We could even say 5' methyl G cap. And then we're going to write the same sequence we have here. A, U, C, U, A, U, G, U, C, C, U, G, G, U, U. But now we're splicing because this is the mature mRNA or the processed RNA. So we want to cut out those lowercase letters, which means that we're going to take this UU and then this A gets brought over next to that U, making that codon. And now we have that A, and then we go G, C, A, C, G, A, U, A, and then C, U, U, A, 3 prime, A, for our poly A tail. Okay, so that's our poly A tail. Okay. Now, it also says, draw a box around the region of the mRNA that contains the coding portion of the gene. And so if we did to do that, we can say the box would be the coding portion. So we're going to just draw a box all the way around our open reading frame. Remember, this would be our 5' UTR. This is our 3' UTR, just to be clear. Okay, and we're just told that 4 base pairs, again, illustration purposes. So this middle part that's box is what is the open reading frame. That's what's going to encode for the sequence that will ultimately the ribosome will read to know which amino acids to put in. Okay, so now this next one is just to illustrate the general way that nucleic acids interact. And what we're asked to do is write the tRNA anticodon sequence. And so what you want to do, whenever two nucleic acids interact with each other, and they interact with each other through complementary base pairing, they're always going to be complementary, meaning an A pairing with a U or an A pairing with a T, depending on if it's RNA or DNA, or a C pairing with a G. That's complementary base pairing. They're always going to be complementary to each other and anti-parallel, meaning one strand, if it's going 5' to 3', the other strand's going to be going the opposite direction. And so when we're having a tRNA associate with an mRNA in a ribosome to read the codon of the mRNA, the tRNA's anticodon, that's the part of the tRNA that's complementarily binding to the mRNA, is going to be in the opposite direction. And so what we want to do is we have, let me just kind of highlight what we have for codons. Here's a codon, there's 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 a codon, and there's a codon. So we need to have tRNA anticodons for each of these. And so what we want to do is we'll then, now since this RNA sequence is going 5' to 3', all of our tRNA anticodons are going to be in the opposite direction. So we can say 3', and then A, it'll be U. So here we have an A, complement to that is U. Here we have a U, complement to that is an A. And then G, complement to that is a C. And that's a 5'. Okay. And so we can keep on doing that all the way through. And so rather than writing 5' and 3' for each of them, I'm just going to write a 3' at this end, and we'll just write the complement of each of those, and then I'll separate them to show that they're anticodons in separate sequences. So here, the U would be with an A. The C and the C would be with G's and G's, and actually we'll just do this just for clarity. That's another codon. And now this next one's UGG, so the anticodon to that would be 3', and then U, complement to U is an A, complement to G is a C and a C, okay, and 5'. And then the next one is UUA, and so again we're going to have 3', we're going in the opposite direction. UUA, the complement of that is AAU. Next is CAC, that's 5'. So 3' CAC, the complement of that would be GUG. Wait, where am I? We have AAU. And then GCA, sorry about that. GCA, the complement would be CGU. And then CGA, the complement would be GCU. 5 prime, and then the last one, we actually don't have an amino acid that associates, or tRNA that associates with the stop codon. Instead, at this point, the stop codon is recognized by these other things called release factors. And so when the ribosome gets to the UAA or the stop codon, whichever one it may be, then release factors enter the ribosome and cause the ribosome to dissociate from the mRNA. And so these would be the anticodons. And now the last step of this first problem is now to say write the polypeptide that would result from this message. So now what we want to do is we're going to use these codons again to use, and this time with our codon table, to know which amino acids we need to put into our sequence. And so we'll bring our codon table over here so that we can see what we have. And now proteins, or polypeptides, have directionality similar to the way nucleic acids do. Nucleic acid directionality, we say 5 prime to 3 prime. With amino acid or polypeptide directionality, we say N terminus to C terminus. So a good way to kind of remember which direction the polypeptide goes, or the protein goes, is the RNA is always going to go 5 to 3, and then the protein goes N to C. And so we're going to have an N terminus. Oops. And then we're going to read these codons here, the AUG, the UCC, the GGU, and then the GGU, the UAG, or the AUG, the UCC, the UGG, the UUA, GCA, CGA. Those are all the codons. That's what will tell what we have for our amino acids. So AUG is always going to encode for the methionine. So we're going to start with a met. And then next is UCC. So we have to look over here on our codon table. And we're going to just look and find UCC is for a serine. So we then go ser. And then after UCC, we go UGG. So we find a UGC, and that's a tryptophan. So that's here, TRP. And then after the UGG, we go UUA. So we then find UUA, and so that's leucine. And then GCA, we then are going to find GCA. GCA is alanine. And then GCA, CGA, we then look on the codon table, CGA is arginine. And then CGA to UAA, which is our stop codon. So we don't write stop, we don't write anything else at that point. So the stop codon, that's where we will then have the release factors come in, cause the ribosome to dissociate, and that's the end of our polypeptide. So that the other end of our polypeptide is the C terminus, or the carboxyl terminus, and so we can just write a C for that. And then the last step of this process, of this problem sheet, is there another gene likely located in this sequence? If so, what is the polypeptide chain? And so what we want to do is look back up here on our sequence, and what we did is, yes, we did identify an open reading frame. We have two open reading frames, the top one and the bottom one. And so the open reading frame, remember, so now for this bottom open reading frame, the bottom strand now is the coding strand, and the top strand is the template strand. And so the bottom strand is what will become the RNA sequence starting at this side, 5 prime to 3 prime. And so what we can do is we can take that sequence and we can write it out so that we know what RNA we're going to work with. And so we would have hold on just a second there. And so we're going to read 5' AUG, CCA, and so on. So 5' AUG, and then we go CCA, CCA, and then we're going to go GTC, GTC, and then TTA. Oops. And this is what you don't want to do, is include a T, because that's a U now, because we're in the RNA. So GUC, so the TTA becomes UUA, and then the TAG becomes a UAAG, 3', and so if we do the same thing as we did in our other one, that is a codon, that is a codon, that is a codon, that is a codon, and that is a codon, that last one being our stop codon. And so now we can again refer to our codon table here and write out what we will have for a protein sequence. And so we can have an N. The AUG is going to be our methionine. The CCA is going to be our proline. GUC will be GUC, valine, and then UUA will be a leucine, and then we have a C terminus, because remember that UAG is our stop codon, and that stop codon again is release factor ribosome dissociates. Okay, so that's how you can go about trying to solve these. Find the open reading frame, what's the resulting polypeptide or protein sequence that would result, how you would handle introns and UTRs and that sort of thing. And so it's in this way that the genome is organized, and we can actually, when we sequence genomes, this is what we look for. When we first sequence a genome, we can scan that genome and try to identify open reading frames. And then we can go and start experimentally determining whether those actually encode for proteins or not. But it is an actual type of analysis that is done routinely in genome sequencing and genome analysis. And so if hopefully this worked for you, and if you liked it, please let me know. And with that, have yourself a great day.